Did you see Rio 3 is coming?
Did you see Neymar tore his ACL?
Oh, that's awful.
I know.
But he's an irrelevant player nowadays.
He's still relevant.
Ah,
Vandry, if the US is playing Brazil at a World Cup
Oh, yeah.
quarterfinals, are you not scared of Neymar?
oh no, I'd be terrified of Neymar. But I
Exactly.
mean, like throughout the year, he might have one or two appearances, right? Champions League is no more
No, he
Neymar.
plays in Saudi Arabia now.
They don't play in the Champions League though.
I know they don't.
Yeah.
He just makes $1.7 billion a year.
You know, if that's the life he
Dude.
wants to have,
Dude, I am so okay with that. Are you kidding me?
dude, you've got to have some passion.
Look, she showed up.
Like Messi should totally go back to Barcelona. Coolest revamp of the year.
Messi, imagine Messi, Suarez, and Neymar all go back to Barcelona. Oh my God.
Neymar's too money hungry though.
They're all
He
too
wants
money.
15
They're
cars.
all too money hungry. It's been recording this entire soccer talk.
Hi YouTube.
All right, let's get started, guys. 7:00 something. We're late, but it's okay. So welcome to topic 5, cell signaling, transduction pathways, by yours truly and Mr. Vandry Sisson.
If you have any questions along the way, just raise your hand.
Feel
and we'll
free
get
to ask them, please. All right.
look at the animations. Don't do them all at once.
Sorry.
All right, cell signaling. Signals are typically small molecules or proteins. They can be hydrophobic, hydrophilic. They can be growth factors. There's plenty of different types. And the typical outcomes of cell signaling are changes in existing protein function, changes in protein levels, and cells don't have to receive one signal at once, right? They can have a bunch going on simultaneously. Crosstalk is a topic that we've talked about in lecture. It's when one part of the pathway communicates with another part that's not directly down the downstream effects. Dude, why is everything animated? I didn't click it. All right, kinases. Kinases cleave off the phosphate group from ATP and attach to the substrate. I like the Khan Academy figure because it simplifies it. This is if you're comfortable with this, you're in a golden spot. It looks like Amoeba Sisters. What? Amoeba Sisters is like has the shading and all that. But yes, that use this figure. Kinases attach ATP, and typically a kinase will attach this phosphate group to one of these 3 amino acids because these OH groups are the great acceptors. So these are the 3 that have free OH acceptors at the end. And that's where the phosphate group will join on. All right, for our RTK, RAS, MAPK pathway function, this is really important for cell signaling, differentiation, and division, and we'll walk through the steps now. Okay, wait, I think there's one. Oh, no. Okay. So the first part is the signal molecule binds, and these receptor tyrosine kinases dimerize, which just means they come together. So they'll start apart and they'll come together. And then they cross-phosphorylate themselves, which means that this guy will phosphorylate this, and this guy will phosphorylate this. So they do it to each other, and that's why dimerization is really important for this pathway. Then once these have dimerized and they're phosphorylated, this protein called GRAB2 binds to the SH3 domain. No, it's covered by Zoom, but it's the SH2 domain. And then this adapter, once this adapter binds, the RAS activating protein, also known as SOS, binds to the SH3 binding domain. You know what PXP motif is that?
Yeah, I have an actual slide.
Oh, okay. We'll go through all these different fun binding domains. And then the binding of SOS to GRAB2, so this interaction right here, brings this protein near enough to the inside surface of the cell to activate the inactive RAS protein. So once we get past here and this SOS, it does its function of activating the inactive RAS protein. The RAS protein becomes active with GTP, and then there's transmission of the signal. The next step in this pathway, yeah.
So in this case, guys, you'll see in the drawing, RAS is attached to the cellular membrane. So it is not moving, it is diffusing through the around the cell. being attached to the membrane.
Yeah, it's got this little like thing that, it's like a roller coaster, it goes around the cell. And then we have this MAPK cascade. And as you can see, these names are horrific. I don't know what biologist did it, but they didn't do a great job. So it goes MAPK3 to MAPK2 to MAPK1. And yeah, this causes the RAS causes the activation of MAPK3 up here, and then it causes downstream effects, and then downstream, and then it can do a bunch of these different functions, right? We talked about all the different ways that it can work. So then, yeah, it activates it through a conformational shift on each. And then there's, as we learned about GEFs and GAPs from last exam, there's a RAS gap that functions to inactivate this pathway if something goes wrong. And it can do its function at any step along the way. Let's see right here.
All right, before we move along, any questions on first pathway? Okay. Oh, cell survival signaling. So, before we get started on looking at the actual pathway, we need to talk about why it's important to have this pathway and why it is widely controlled by the cells. So, the default for any granted cell in your body is for it to die. They are programmed to die because not one cell is supposed to live forever and perform its functions. As you know, we age, as things change, the cells are meant to be dead at the end. So this pathway specifically inhibits apoptosis, which is programmed cell death, which is a process that the cells can go through to prevent any sort of mutations from spreading. And so every time you get a sunburn, for example, your cells, when you start peeling, that's because your cells committed mass programmed cell death to prevent that from affecting the further cell lines. So why else? Occurrence of crosstalk with other pathways. At this level in this class, like we don't really talk about the specific proteins and their functionality like elsewhere. But just know that this pathway specifically talks around with a whole bunch of different pathways. These proteins involved in this pathway are involved in many different other very important pathways, like mTOR, PK1, P1, PI3K, like they all are very important. So first step is the survival signal is going to bind to the RTK. As Van was talking about, RTK stands for receptor tyrosine kinase, and they have kinase activity. So second is the RTKs are going to dimerize, and they are going to cross-phosphorylate. So they will be active because of each other. And then P13 kinase is going to bind the phosphotyrosine site via the SH2 domain present on the RTK, or it's going to bind to the RTK because of the phosphotyrosine, because it has an SH2 domain. And so P13 kinase or PI3 kinase is going to phosphorylate that little molecule called inositol phospholipid. It's called P13 kinase because you go from a P12, which is a diphosphate phosphoinositol, to a triphosphate phosphoinositol. And so very important here, the inositol phospholipid or triphospholipid, whatever you want to call it, is very important because it'll act as a binding site for the next proteins in the pathway. So it becomes a binding site for PK1 and for AKT. I'm gonna activate everything. So PK1 with a pH domain. pH is the domain that binds to the phosphoinositol. Also, write these domains down. You might be asked in the exams. I can't guarantee you that you will, but I also can't guarantee that you won't. So write them down. Write down what they are supposed to do. We'll also cover them in a second. So PK1 is going to bind to PIP3. And then AKT also needs a PIP3 available because they both bind to that. Similar to RAS in the previous pathway, PIP3s are going to be stuck on the membrane. They're going to diffuse around, stuck to it. But upon the PK1 phosphorylating AKT for the first time, then it causes a conformation of shift on AKT. When AKT has this conformation of change, then it becomes available for protein kinase 2 or mTOR, whatever you want to call it, to phosphorylate it. And so here you'll see that AKT gets phosphorylated twice. Once AKT gets phosphorylated twice, it becomes active. This activation causes a conformation of change, which then causes it to diffuse away from the PIP3. Once it is active and diffused away from the PIP3, it is going to go on and phosphorylate the protein called BAD. Why is this important, John? Well, BAD is the protein that sequesters a protein called BCL2. That will be the protein that prevents apoptosis from happening. So basically, this is like a counterintuitive mechanism because you are phosphorylating something to inactivate it. So BAD needs to be phosphorylated to be inactive. BAD is normally active without being phosphorylated, and its function is to hold on to this protein called BCL2, the red protein on the screen. And so once it is phosphorylated, then it changes conformation, which makes it lose affinity for BCL2. And with this BCL2 being free, then apoptosis is going to be inhibited. BAD and BCL2. Like I was talking to you guys about this, apoptosis means programmed cell death. BAD is the protein that activates mechanisms for apoptosis by holding onto BCL2, as far as this pathway goes. And BCL2 is a protein that inhibits apoptosis. So there are two possible scenarios. So active BAD will be bound to BCL2, which means the BCL2 will not be active, which means it will diffuse through the mitochondrial holes and not diffuse through the mitochondrial holes. The mitochondrial holes will not be patched, and then apoptosis will be programmed. Do not worry about the mechanism that leads to apoptosis. Just know that BCL2 is the one that is inhibiting it or not inhibiting it, depending on if it is free or not. So inactive BAD will be unbound from BCL2, which means BCL2 can be active, which will then lead to the inhibition of apoptosis. Is there like a full picture? Hold on. Okay. So, sorry, there's no full picture. AKT needs to be phosphorylated twice. Once it's phosphorylated twice, then it diffuses away from PIP3. Once it's diffused away from PIP3, then it's going to look for BAD. Once it inactivates BAD, then apoptosis is inhibited. If it never is able to phosphorylate BAD, then it will not prevent BAD from sequestering BCL2, which will then lead to apoptosis. Did I go too fast? Does anybody have any questions? Okay. So this is just a summary. We just went over this. It's more for studying. Oh, yeah, there was a picture. Okay. But so recap, survival signal binds, RTKs cross phosphorylate, P13K binds, becomes it activates and then it phosphorylates PIP2 to PIP3. You need two PIP3s in order for the PK1 and AKT to bind. PK1 is going to phosphorylate AKT once, then mTOR is going to be able to phosphorylate AKT. AKT will then dissociate away, and then it will inactivate BAD. All right, so now our third pathway, JAK-STAT. So we have a breakdown of the pathway coming soon. So this one's a little different. So for the two previous pathways we're looking at, the receptors were able to cross-phosphorylate each other. For this pathway specifically, they are not. Oh, John, why does this matter? Well, it matters because once the signal binds, they need something to cross-phosphorylate so they can dimerize, right? And so what does that for them? Kinase is called JAKs. So JAK1, JAK2, once the receptor binds the signaling molecule, which is in this case prolactin, the JAKs are going to cross-phosphorylate each other, which will then cause the tires or the receptors to dimerize. Once the receptors are dimerized, then JAKs are going to phosphorylate the receptors themselves, which will then allow for the STAT molecules to bind to the receptors. And so once the STAT molecules are bound to the receptors, then the JAKs are going to phosphorylate them again, which will cause conformational shift on both of them. This will lead to them losing affinity for the receptors. And once they lose affinity to the receptors, they will be diffusing around the cytosol looking for each other or another molecule of the same kind so they can dimerize. Once they are dimerized together, then they are able to diffuse or to go into the nucleus with the use of important, and then they serve as transcription factors and upregulate or downregulate the transcription of milk protein genes. Did I go too fast? No? Okay. Yeah, this is no. Yeah, very crucial here. I know I talked about it briefly. JAKs are not RTKs. And then the receptors are also not RTKs. So neither of them are RTKs. For these receptors specifically, they don't have kinase activity, which is why the JAKs are there. And scientists are very creative. JAK stands for just another kinase. Any questions? Okay. Oh. That's just a breakdown for afterwards. Okay.
All right. Last but not least, we've got GPCR pathway. So this figure on the right is probably what you want to put on your note card. It's the only figure I think Laura gives in her lecture notes, and it's really important, so we can just walk through it. The 7 transmembrane receptor bound is this little squiggly line in black. It goes through 7 times. And it has 3 subunits that are typically attached to it. It's got the alpha, the beta, and the gamma. The alpha in this image being the blue, the beta, the red, and then the gamma is the green. And so the alpha, the blue part, is a GTPase. So it takes GTP. Its typical function is taking GTP and converting it back to GDP through hydrolyzation. So this is the inactive state in the top here, right? Nothing's going on. They're just all bound to one another. And in this picture, the blue is attached to GDP. So then if we're moving to the right, we've got a ligand comes in and binds to the receptor. And that makes this conformational shift in the receptor. And then it causes G alpha to let go of its GDP. And then GTP floats in. Once that happens, it causes another conformational shift in G alpha. And the G alpha then dissociates from beta and gamma, as we can see here. All right, one more. And then once this G alpha is bound to a GTP and it's let go of the beta and the gamma, it can activate a signal transduction cascade or an effector in the downstream for cell signaling. Once that signal has been given, we need to turn it off. So there's this protein called RGS, which is this light blue over here. And what that will do, it will accentuate the GTPase activity of G alpha. So G alpha is doing its function, and this RGS is saying, okay, you need to do your function faster. So it will turn that GTP into GDP. And then once you have GDP back here, we're going to go back to the center, right? Because we started with GDP bound, and everything comes back to one unit, and it waits for the next signal or ligand. So this GPCR up here is a guess analog, right? Because it turns GDP into GTP, or it doesn't turn it, but it facilitates that process. Whereas this RGS is doing the activity of a gap, where it's turning GTP into GDP through hydrolyzation. Here's another image that represents it. Again, same process we've talked about. Another image. I put a problem set question. Oh, it must have not reloaded. We can talk about it later. Okay, yeah, go on.
Okay. So I want to just highlight a couple of common misconceptions. So first one, phosphorylation is a covalent post-translational modification that is happening in proteins. So very important here, kinases are going to phosphorylate the hydroxyl groups in tyrosines, serines, and threonines. Phosphorylation is not an ionic interaction between 2 charged amino acids, or between the charged amino acid and between phosphate. This is a common thing that is said on exam questions when asked. The phosphorylation event is going to happen where a kinase is going to grab a phosphate group from either an ATP or GTP, and then will deliver it to the accepting hydroxyl group on one of those 3 amino acids. It is not going to be an interaction between a positively charged amino acid and a phosphate. That is a common mistake on exams. That doesn't exist. Another thing, SH2 domain is a phosphotyrosine binding domain. John, what does that mean? You have a phosphorylated tyrosine, protein is going to be able to bind it through that. SH3 domain binds a PXXP region of a protein, PXXP motif, whatever you want to call it. So PXXP means proline rich. So what this means is it's literally a region full of prolines. John, why does that matter? Well, so the drawings over there exemplify it good. So you see how there's a whole bunch of alpha helix on one part of the protein, and then there's a bend. Commonly on like ribbon style models, you'll see like those bends. Those are proline rich regions. And why that happens is because, as you might remember or not, prolines are the amino acids that form a cyclic ring with the R group. So they are not able to hydrogen bond. Because they're not able to do that, the helix structure cannot be formed with prolines. So if you have a region full of prolines, you will not be having alpha helices. So that's what it looks like. The SH3 domain is going to go in and bind that specific region. And so the last domain I wanted to talk about is the pH domain. And it's the domain that binds the PIP3 protein. I've only seen that be asked once on exams, but I just wanted to let you guys know so that you're not caught off guard by it if it is asked. You can talk about. You want me to reload the page?
Oh, no, I think I just need to go out of the presentation. All right, I have one problem set question I'd love to go through. All right, if you've already started the problem set, you've probably have an answer or a rough draft of an answer. Would anyone like to share? I'll read the question. GPCRs activate G proteins by reducing the strength of GDP binding, allowing GDP to associate and GTP, which is present in much higher concentrations, to bind. What would happen if the affinity of G alpha for GDP were reduced, but the affinity for GTP remained unaffected? Explain your reasoning. Make sure to explain the effect on the amount of pathway signaling as compared to the non-mutated G alpha, and then why. Does anyone have a guess? Here, I'll bring back. So what the question's asking is, what if this G alpha really couldn't bind to GTP, and right, we're inactive when we're bound to GTP. What happens if we could only bind GTP? Yeah.
I'm just say active.
Heck yeah. Yep. Oh, dang it. Why doesn't it animate the one slide I want? Yeah. So typically GDP is tightly bound to G alpha until it's stimulated by the GPCR receptor. But with our mutant, it'd be constantly active, right? Because this G alpha is constantly rebinding the GTP. So even when RGS causes the GTP to hydrolyze, G alpha will immediately dissociate from it, and then GTP will come back in and it'll be active. So make sure to mention that the pathway is on even when the signal is not bound. And then I think that's it for our slides, right?
Yeah, that's it.
All right. We are happy to stay for the rest of the hour. If you want to work through problem set questions, if you want to head out, totally get that. These slides will be sent to you and the recording will be sent as well by Zoom.